Thank you all for joining us today. It's the last of our 2026 Pendleton Science Roundup seminar series. And this is something we plan to do each year. So if you're interested in presenting next year, please reach out. I'm also looking at getting these posted online. We'll have this sent out in email chains and distribution lists, but right now we're planning to have them announced and posted on the USDA Pendleton ARS site. So please check back. I think it'll be another week until we're able to get them posted. So today I'd like to introduce to you Tom Ducey. He's a microbiologist in South Carolina, and he's been with the agency about 21 years. His focus is on ag products and soil sustainability and collaborates a lot with NRCS and the EPA. So thank you very much, Tom, for joining us today, and we look forward to your presentation, taking control of your soil's health, influences of amendments, biologicals, and managements. Thank you, Tom.
All right. Thanks, Kate, for the introduction. Thanks, everybody, for attending today. So I'm going to talk a bit about the work that we've been doing in the coastal plain here in the southeastern United States. So I'm based out of Florence, South Carolina, and I have worked here for almost going on 20 years. I worked with the USDA in Peoria as a postdoc working in food safety. So it was a bit of a switch, but I'm happy to say that things seem to be going pretty well here in South Carolina and have for the last 2 decades. And so when I was brought on, a lot of my work early on was looking at waste treatment development, and then that slowly morphed into recycling nutrients and reuse, and then how that impacted the soil microbial ecology. And then it transitioned, especially in the early 2010s when soil health picked up and NRCS had stood up their soil health branch. That soil health became a major focus of my program, and it has been ever since. So as Kate mentioned, my title says we're going to talk about amendments, biologicals, and managements, not necessarily in that order, but if I time my talk correctly, we'll get through all of that in time. Now, one of the reasons that soil health is such a concern is that roughly half of all farmers do talk about having degraded soils to some degree. So at least 50% of farmers have at least one concern. Typically, these are erosion, soil compaction, poor drainage, and low organic matter. Those are all very common concerns. And so, we look at how we can address these. And the good thing is that these issues can be addressed if we can properly manage the soils. And we do have a number of tools that can be brought to bear. Why are soils so important? Because they are essentially, and as a microbiologist, I put it into the context of soil is a living ecosystem. And it is, per 1 gram of soil, there's roughly 1 billion microbes, and they do various functions to provide system ecoservices to their environments. And so they sustain plants, animals, and the humans that subsist off of the land. And so when we have a healthy soil, typically what we can expect is that we have great water quality and air quality as well, actually, and that these soils will retain nutrients and water, and in turn that retention can provide plants with a healthier life cycle. And then also we should see that hopefully in yields as well. And I will touch on yields because that is a interesting subject in and of itself. But how do we determine if a soil is healthy or not? We have developed a number of tests, and I'm sure that the people that are listening today know some of them. We'll touch on a couple that we have used and that we report on fairly often. But these can be broken down into a series of biological, chemical, and physical properties, and we just refer to them as indicators. And then we can track these indicators over time, and they let us know how the soils are then functioning. So if we change in management, do the soils improve in their functionality, or do they actually degrade? I always tell people that are adopting soil health uh, indicators and they're tracking those that it's a good tool to use if you're changing soil management. So if you're living in the southeast coastal plain and you say, well, I want to look at my soil health, and you compare it to the Midwest, you're going to be shocked by the numbers because these tests, certain indicators are different across regions. And so keeping it within a manageable context. So basically, how are you changing your soil and how is it responding to those changes is a really good way to use soil health tools. Now, the Soil Health Institute, which is based in Cary, North Carolina, has done a lot of work at looking at these soil tests and which ones seem to play an outsized role in driving soil health. And they have come up with three: aggregate stability, total carbon and nitrogen or organic matter, and then soil respiration as the three big drivers of soil health. So, as I mentioned, two of the soil health tests that we use a lot, and there are many more, are SMAF. SMAF was developed by ARS back in the early 2000s, and that stands for the Soil Management Assessment Framework. And as I mentioned, it is like CASH, which was developed at Cornell University, which is a comprehensive assessment of soil health. They are broken down into physical, chemical, and biological indicators. And then the green arrows just indicate the ones that the Soil Health Institute marks as being the biggest drivers. Now, it should be noted that SMAF was actually developed and trained on soils in the Midwest, and CASH was developed and trained on soils in the Northeast. And so when you are a farmer in the Southeast or even in the Pacific Northwest and you use these tools, you can expect that they will not align with what is a traditional healthy soil, just given the data sets that these were initially trained on. Though there are efforts now to bring these, to train these across the United States to give us a better representation of all agricultural soils and not just those in the regions that they were developed. Now, as I mentioned, I am in the coastal plain, and that is NRCS's major land resource area, 153A. And 153A is indicated, and basically it runs down from Virginia down through North Carolina, South Carolina, and Georgia. And so if you're familiar with the U.S. highway system, basically everything east of I-95, when you look at the soils, you can assume that they're coastal plain, and compared to everything west, which is our other MLRAs. There are other very adjacent soil types that extend from Texas through the essentially the Bay areas surrounding Louisiana, Mississippi, Alabama, and Florida as well, and then up into the Chesapeake area. But these soils are weathered, they're degraded, they've been farmed for centuries at this point, and so they are characterized by having low pH, low water holding capacity, and this is predominantly due to having essentially a low carbon content. And so overall, our soil fertility compared to other areas of the United States is much poorer, unfortunately. A typical soil in the MLRA 153 is Goldsboro. As you can see, it's predominantly sand. And then we have it's acidic, pH roughly 5.8, and then about 0.5% of organic matter, which is not great. So we have spent roughly, the center was stood up 61 years ago, and we have studied how to improve the coastal region soils ever since then. And we've looked at a lot of different managements and soil amendments. I would have put kitchen sink up here if we suspected that it would actually help improve soil health, but it does not. So everything but the kitchen sink is something that we have studied at some point or another. I don't have time to talk about all of them, but I will talk about the ones into certain depths of certain degrees here in this talk. Basically, conservation tillage, biochar, which is we've been known for roughly the last 2 decades of research in, and then insect frass, which is a new one. And then I've also been told that there is a good degree of microbial interest in microbial inoculants. And since we do work in that area, I thought I'd talk about that as well today. Now, conservation tillage, as I mentioned, we have we have a series of plots that were stood up over 40 years ago in conjunction with the Department of Energy when they wanted to look at stover removal and how that would impact soil fertility. And so that study has been ongoing ever since then. And I wanted to take this slide to talk a bit about when we talk about yields. And so we know, and you can see by this in the top graph, that when we look at conservation tillage, when we add cover cropping, we can actually see an increase in microbial activity. So cover cropping does seem to boost microbial activity. When we look at it in conventional tillage, cover cropping doesn't seem to move microbial response at all. But then when we look at yield, and this is in cotton, those arrows point out. Now, one of the other indicators or properties of soils in the coastal plain is that we have very low electrical conductivity. And so when we look at yields in these areas, they tend to be much lower than areas that have what we would call medium to higher EC, though the ECs, even our high ECs in our area, are much lower than you would find in other parts of the country. Now, if you actually grow the cotton under conservation and cover cropping managements, we're actually able to bring the yields up. So the mean. So if we look at in the middle, the brown one, the brown area of the graph, that's roughly that's 6 pounds per 10-foot row of cotton. But when we actually introduce conservation tillage and cover cropping, we actually get a 33% yield increase. So when people ask, can these managements improve yields? In a lot of cases, the highs are always going to be, there's a ceiling. So in good times, when your high is high, that's what you can expect. But when you have either regions of your farm that will stress your plants out because of soil conditions, or if you find yourself in climatic events where your plants are stressed. When you have these additional managements or amendments, your high will always stay your high no matter what. But during these stressful times or stressful conditions, your yields may not be as low. So it'll actually bring up your low yields. And so over time, you can say, yes, we do get improvements in yield, but it's not necessarily how we think of, when we say, okay, we got increased yields, you're going to see it all the time. It's really only in certain conditions, which is not usually what people want to hear, but the science does support this model, at least in the southeastern area. Now, when we looked at the accumulation of carbon over time, conservation tillage actually gives us almost 2 times the amount of soil organic carbon compared to conventional tillage. And this is significant. So we go from around 8 to 9 grams per kilogram of soil up to around 16, 17. So almost a doubling. Unfortunately, we have noticed that you get a rapid gain of soil organic carbon in the first 5 to 10 years of this practice. However, it then starts to smooth out. We plateau, and by the time you're 40 years in to this study, we are not getting any more stored soil organic carbon. So the outputs, we gain them and then we lose them at the same time in that same year. And so that's either to microbial mineralization or more often leaching. And so at this point now, we really need to develop new treatments and practices that can raise the ceiling on these saturation levels. And so a lot of the work in our amendments are in that vein. So 3 of the amendments that we are looking at to varying degrees are biochar. And biochar is basically taking organic material. We try to valorize animal waste, agricultural wastes and byproducts into a stable carbon-rich material. under pyrolysis conditions, which is high temperatures, anywhere from 350 to about 950 degrees C, and low oxygen conditions. We've started looking at frass, and frass is basically insect manure. Um, so we try and treat it just as we would any other animal manure, though frass does have chitin in it, which does actually play a role in plant protection to varying degrees. And then inoculants. And so inoculants are primarily microorganisms, bacterial or fungi, that can act as biostimulants. It goes under a lot of names to promote soil or plant health and hopefully boost yields. Now, biochar, as I mentioned, the Florence area, the ARS Center, has looked at biochar for almost 2 decades at this point. And we have identified that you can actually, by manipulating either the feedstocks or the pyrolysis conditions or blending different feedstocks, you can actually design, so we call it design of biochars, design the char to address specific physical or chemical deficiencies in your soils. And so we know which chars to use if we want to increase nutrient retention, reduce or increase water holding capacity. We want to increase soil aggregation or reduce bulk density and they all do a fairly good job of increasing carbon storage. And then, as Kate mentioned, I have worked with the EPA to use agricultural-based biochars. Manures in particular are very good at sequestering heavy metals and other contaminants of environmental concern. And I'll actually talk about that in the upcoming slide. And so together, we can take these biochar properties and use them to hopefully increase soil health over long periods of time. So as I mentioned, we are working with Western North Carolina. At this point, Western North Carolina, as those of you know, we're hit very hard by Hurricane Helene in 2024. And so they are still digging out of the aftermath of that. A lot of their soils were contaminated. I was actually up there yesterday talking to farmers who said as they stood out and watched the floods, everything from oil tanks to propane tanks to cars were floating by and over their fields. So you can just imagine the level of contamination that was occurring. And so some of these fields, the notes from NRCS were, this has been seeded 5-6 times and we still haven't gotten anything to grow. And so we're working with NRCS and North Carolina Department of Ag to put into effect NRCS Conservation Practice 336, which is soil carbon, and using biochar as the method to achieve that. And so here is one of the carbon pyrolyzers that is performing that work, making the biochar, and there's roughly over 1 million acres of damaged forest land, which actually poses a significant wildfire hazard to the region. And we're cutting down those trees, turning it into biochar, and then giving it to the agricultural producers in the region. And so this char does meet the NRCS organic carbon thresholds, and as well as the hydrogen to carbon organic carbon molar ratio. And then looking at heavy metals, they are very low. So we don't run the risk of contaminating the soils further with arsenic or cadmium or lead, because these all meet the NRCS method. So the char is good. And so they have started to go ahead and actually use equip money provided by NRCS to put this biochar into the ground. And so we did a quick pot study with several of the soils. And we wanted to look at basically how we could move soil health metrics in these soils. So this first slide basically shows that when you increase the amount of biochar in these soils, the first thing that we see is that we can actually increase the field capacity. Western North Carolina can get hit with drought-like conditions over time. And so biochar actually is a very porous material. And so it actually does a good job of holding extra water. And that, as you would imagine, does increase with the amendment rate. And so this will provide a period not too long, but it will provide 4 to 7 days of additional water for the plants when they do find themselves in drought-like conditions. Conversely, it also reduces bulk density. So bulk density basically tells us what is the soil's resistance to compaction. The lower your bulk density is, the less likely it is to compact. Lower compaction means you get better root penetration, better aeration of the soils, and and it actually improves drainage after flooding. So if one of these 1,000-year floods occurs sooner than the next 1,000 years, those soils should drain more quickly. And it seems a little bit counterintuitive. It holds more soil, so these soils hold more water, but they also drain better. But you know, knowing that the biochar is sort of like a sponge, and so once it absorbs as much water as it can, the rest just flushes out. It does. It starts to make a little bit more sense. Or maybe I'm the only one that has that problem making that connection. But additionally, we have a long-term study looking at biochar here at the center. In this one, instead of using hardwoods like we are in Western North Carolina, we looked at pine chip and poultry litter. As I mentioned, poultry litter is a pretty good char, especially if you want to ameliorate soil contaminants. And then we looked at a blend. As I mentioned, our soils are very acidic, so using these as a liming agent works really well. And then actually poultry litter is pretty good at providing P and K as well. So some of these chars can be used as a fertilizer of sorts. Again, like with the previous study, we are able to increase soil moisture with these biochars. Now, pine chip works much better in this regard than poultry litter or the blend. So the woods, hard and soft woods, are known to have very good water holding capacities. Grasses as well, so switchgrass, miscanthus, and feedstocks of that nature are known to do very good with water and nutrient retention. Likewise, bulk densities, in particular the pine chip and the blend, do a very good job of decreasing bulk density. And in the Southeast, that is very important because we have a very shallow hard pan. So if we can get this char down through that clay layer, it does a really good job of getting root penetration. There's nothing like growing your corn and then a hurricane comes in, because we do get a fair amount of hurricanes here in the Southeast, and seeing all your corn blown over, and then you lose the whole crop. So having biochar in the soil to alleviate that compaction and getting those roots down deeper is very beneficial. And then, I just wanted to show a couple of those, but here is a Venn diagram looking at pine chip, which is in the left side, poultry litter, which is on the right side, and then the blend, which is in the middle of that Venn diagram. And we can see that after 8 years, we move a number of these soil health indices towards the positive over long periods of time. The poultry litter, it's more in the chemical side, so looking at available P and K, and then the pine chip does a really good job at moving those physical characteristics, though there is a boost in soil respiration and then soil organic matter as well. And that just goes because poultry litter is about 33% carbon, where pine chip is roughly in the 80 percentile of organic carbon once pyrolysis is over. I'm just going to briefly touch on frass. Frass, as I mentioned, is insect manure. It's essentially after you grow, and 3 common sources right now are cricket, mealworm, and black soldier fly. A lot of companies in the Southeast, it's EnviroFlight. Tyson Poultry is also looking at frass as well, but it's the bedding that the material the insects are grown on, their waste, and then when they shed. So, and those exoskeletons do provide chitin. And that's important because when we looked at this was a study that was done by Amanda Ashworth in Arkansas, ARS location in Arkansas, that high frass rates were able to resulted in lower leaf damage compared to control, so no frass or poultry litter treatments. So we do get an increase in leaf resistance to insects, to insect damage. And then those high frass rates actually resulted in a 2 to 4 time increase in nutrient use efficiency. And so this is an ongoing study, and we are being rolled into this study as well, where we're going to establish plots in this upcoming year, looking at sweet corn. And so here in Florence, we're going to be looking at soil health improvements and plant resistance to pest damage. And lastly, I'm going to talk a little bit about what I refer to as the wild, wild west of agriculture at this time, and that is biologicals. What are biologicals? They're simply beneficial microorganisms, as I mentioned earlier, bacteria, fungi, that are applied either to the soil directly or plants, and that can be either at the seeds, at the root zone, or foliar, in an attempt to improve productivity and crop health and soil health. Now, these are often marketed as able to, and they're marketed for a wide range of uses, but predominantly it's to enhance nutrient efficiency, increase stress tolerance, and or improve crop quality. And I call them inoculants, microbial inoculants, but they've been referred to as bioactivators, biologicals, biostimulants, phytostimulants, plant growth enhancers. They go by a wide range, but typically, if it contains microorganisms, it's an inoculant of some form or fashion. Now, a little bit more inoculants, they typically come either dry or liquid. That's really the only two ways that they can come. And they are in formulations using what is typically referred to as a carrier medium. And we'll talk a little bit about that in the next slide as well. The dry carriers are typically anywhere from peat to perlite to talc or charcoals. And then the liquid carriers tend to be predominantly water, and then supplemented with either sugars or humic acids or oils or other polymers. Now, I should mention that, a lot of these carriers, they say, well, they're inert and they're just to keep the microbes alive and happy. But some of these actually do have plant stimulating capabilities. The humic acids, molasses, glycerol can all stimulate either plant or microbe growth and activity. So this is what a typical, if you buy one of these products, this is the information that should be provided on the package. It's just like if you were opening up a can of corn, it would list your ingredients, the vitamins, the minerals. And so the top in this example is the ingredients that the product contains. So in this one, it was a mix of water, molasses, amino acids, humic acids, kelp, and fulvic acid. And so molasses is high in calcium, magnesium, iron, and potassium. So that can stimulate plant growth. Molasses is a sugar, and I know in Kate's talk, we talked about sugars and how that can stimulate plant growth as well. And then at the bottom, you'll have your species listed, and typically it'll be reported as a CFU per gram, and CFU is just colony forming units. So if you culture these out on a petri dish, and you took a gram of that material, so roughly about 1 mil, that's how many organisms you would expect to grow on that plate. And so in this case, you would expect anywhere from 1.4 million to 3.9 million organisms to grow on those plates between those two organisms. Now, I always recommend that people spend some time really looking at that information because the devil is in the details. And so if the product label doesn't have a lot of this information or they're very nebulous, I think that that's rather telling. And I'll leave it up to each individual user. But if they're not very forthcoming on what is contained inside the product, hesitate using it. First, your soil health, but then I'll talk about towards the end. So, do they work?
Hey, Tom, your
Oregon Department of
internet.
Ag, they actually were at,
Oh,
it's wigging out.
yeah, if you can maybe
I
turn your
apologize.
video
Hopefully
off.
everybody can. Okay. How do I do that without all right.
Thanks.
And she had mentioned that or Department of Agriculture in Oregon was actually standing up a microbial analysis unit as part of the fertilizer program. And so 2015 was one of the first years that they did this, and they actually tested 27 biologicals, and 24 of them failed. And I was trying to be very generous with what I considered failure. And I said if the product had at least 50% of what was on the label guarantee, we considered it a success. And only 3 products actually passed that criteria. Actually, the Department of Ag in Oregon has actually increased their acceptance rate, so their pass rate, as greater than 80%. So in that case, 27 products would have failed that year. So as you can see, not a lot of the biologicals had a good start at the start of this program. So have things gotten better? Well, in 2022, which is the last year of reported data, publicly available data, they tested 17 biologicals, 13 failed. 10 of them failed entirely, so nothing grew at all. And then 3 partially met their label guarantees. And then hitting that threshold, and actually have 80% here, but it was actually 85%. 4 of the products actually passed the label guarantee, and then 3 partially met that label guarantee. So 7 years later, not really the best of success. Now you can actually go, all this information is publicly available. So if you want to go to the Oregon Department of Ag and go to their fertilizer program and look at their reports and publications, you can actually find this information. So it's all publicly available, what products were given for testing, and then the results that were found by the fertilizer program. Now that's just talking about whether these products actually had organisms that grew. And so Cotton Incorporated decided to test a series of products over 2023 and 2024. And so they looked at a lot of the extension across the US and the Cotton Belt. And so they did a multi-state product testing. And so they actually provided me the products to test for both composition. So what did the microbial communities actually look like and whether they actually grew. And so if you look at the lower right-hand side of this slide, you can see that they all grew. I didn't test product 6 growth because it was fungal. And I'm more of a bacteriologist than I am a mycologist. But all of the microbial products, the bacterial products grew just fine within the stated amounts that were reported on the label. When we actually went and sequenced those organisms, though, it was an entirely different story. So all the single species products, when we sequenced them and looked at the relative abundances, they all matched what they said on the label. So, 97, 95, 97% of the organism that the company said was in the product was there. When we started getting into these multi-species products, that's when things got a bit problematic. One of the products said there was 800 different microbes and fungi in it, and we sequenced 14. Another one said there was roughly about a dozen, and we sequenced like 20, a couple dozen. And then one of them also had a human pathogen in it as well. And whether that was in transfer from the location that bought the products and then distributed it to the multi-state participants, or whether it was a failure of the product's QA QC, is open for debate. But it does go to show that if you're not careful on handling these products, you can actually introduce pathogens into this, and then when you're handling it down the road, it can cause significant problems. In the upper left-hand corner, this is how the products performed compared to traditional amounts of conventional fertilizer. And it's pretty clear here that even looking at lower amounts and cotton is typically fertilized at a rate of about 80 to 100 pounds of N per acre. Though really at 40 pounds per acre, you do get pretty good yields, but none of these products actually led to a significant increase in yield.
Tom, quick question
Yes,
on the products. Did you also add inorganic nitrogen on those?
yes, yes. So those were done at I believe the 40 pounds per acre
Okay.
amount. Though
Um,
I've slept since then, so that I think we did add a little bit of N just to ensure that that we would get things to grow to a suitable degree.
okay. You also have a question in the chat.
Sure.
Often biostimulants are applied at 1 to 2 gallons or less, I assume an acre. So nutrient levels are trivial. What else is a benefit?
Benefit of actually providing
I
the
think,
microbial inoculants to the soil or to the plants.
yeah, if it's added at 1 to 2 gallons, do you think that there is a
Yeah.
benefit?
Well, now I'm going to say I'm a microbiologist, so I'd love for these things to work. And I mean, some of the organisms, some of the products actually say, well, it'll help in plant resistance to rusts and wilts and things like that. So it provides a barrier. Um, some of them will actually help nutrient recovery, or some of them have said these are nitrogen fixers, so you can actually drop the amount of nitrogen that you fertilize with.
Mhm.
Unfortunately, one of the major products that actually touts itself as a N fixer, a free living nitrogen fixer, so it doesn't have to form a symbiotic relationship with the plant, actually doesn't have the nitrogen fixing genes in it. So if you actually sequence that organism, the nitrogen fixing genes aren't there. And they've spun it as saying, well, it actually works with the other soil's free living nitrogen fixers to then stimulate nitrogen fixing in the soil to then boost the plant. So it's a bit of a mixed bag. You add so few of these organisms in the soil at any
Mhm.
single time that I do wonder whether these can actually are providing um, any benefit at all. A lot of the instances where we've seen good um, responses to using inoculants is when we're actually targeting pests. And so you don't need a lot of the bacteria on the plant to be ingested by the invasive pests, but then they get to work growing inside the host and then helping curb uh, invasive damage and things like that. When we actually get to the crop production side, that's where things tend to die off in terms of actually getting good yield, good reproducible yields. And then sometimes the producers actually turn around and say, well, it's because these products are really good if you apply them under stressful situations. So when we have, things like drought or very low nitrogen, then these products tend to to shine. Unfortunately, nobody knows when droughts are going to occur. So saving these organisms for those instances is hard. And then no one that I know is really going to run around and not fertilize their crops to an appreciable degree. So unfortunately, it's a bit of a mixed bag with these.
Thank you.
And then lastly, you know, if even if I say if you're going to buy the line that these can help increase yields, there are a lot of things I think that need to be considered prior to using them, though. As farmers, you all know that you're not just working as a farmer. You've got a lot of hats that you have to wear. Everything from predicting markets, uh, financials in your business. So you've got you have to take business considerations, farming, agricultural considerations. Um, you know, and now if you're going to start working with biologicals, you need to become a part-time microbiologist as well because you have to handle these things, and I'd handle them with a degree of caution. As with everything that gets applied to your field, they can be aerosolized, so they do pose an inhalation risk. And some of these microbes, if they're not overtly contaminated with a human pathogen, which if you handle it, you could wind up with an infection. Some of these microbes that are contained could be opportunistic pathogens. So they tend not to cause disease in healthy humans, but if you are immunocompromised, you do run a risk of contracting a disease. And there are a number of ways that it's like, well, who's going to be immunocompromised? Well, pregnant women, newborns, people that have diabetes and autoimmune disease, and cancer survivors and transplant recipients all have immunocompromised, can all be immunocompromised. And my immunology professor in college was always fond of saying that the immune system was there to get us to the age where we can reproduce, and then after that, all bets are off. So as we age, our immune systems actually do drop as well. So these are things to be cognizant of. And then lastly, there hasn't been a lot of research looking at when we add these microbial inoculants, are we kicking out beneficial bacteria that are already there? So ones that are already providing ecosystem services to the soils, and they're native. They have grown along these populations. They have adjusted to your managements over decades. And so, when you add these organisms, are you going to disrupt those food chains and those ecosystem services in place of these new organisms, and which could potentially, over the long haul, damage your soils more than help them. So a bit of a Debbie Downer, I'm sorry, on the biological inoculant front. So in conclusion, soil health is an excellent tool for tracking long-term progress of your managements. There are a lot of managements that can be brought to bear in improving your soil's health and fertility. A lot of them are impacted by regional availability and land management practices and your soil types, but they can be designed to address specific soil deficiencies and your goals as a manager and steward of your land. And then we just need to know that there are a number of factors, both soil and environmental related, that can potentially limit the overall effectiveness of certain treatments, and essentially no one size fits all. So I appreciate everybody's attention today. I think we have a little bit of time left for questions, but feel free to contact me if you have any further questions. And if you can't get in a hold of me, Kate, I'm sure, would be happy to relay that information or answer those questions directly herself. So with that, if anybody has any questions, I'd be happy to talk for, I think, the 1 or 2 minutes we have left.
Yeah, we have a couple of questions in the chat. So thank you very much, Tom.
Sure.
There is, this is from Francisco. You make a good case for the lack of response with the products you tested. Are there any inoculants out there that showed field scale evidence behind them? Or which biostimulants could be successful?
So to date, in the systems that I study, I have not seen anything that I would say, yes, this works. And I know Cotton Incorporated has put a lot of money into this. FR is actually standing up a program here to look at inoculants, and so I'm hoping that within the next, 4 to 5 years, we'll start to see products that can be used in broad scope um, across the country if there are any. Right now, though, I really haven't seen anything in the systems uh, that we study.
Okay. Um, oh, Dave, or I think someone else is
Yeah,
trying to speak to.
yeah. I was just going to share. There was a workshop on Monday, I think led by Lisa Foltz, that brought in a person from California who works in the department that assesses what I think they call them biostimulants, which includes everything you shared about Tom. But they've developed a list, but I don't think the list of is actually of ones that have efficacy, but ones that when they test them, they meet the product label. So your question, Francisco, is a good one. How do we know which ones really work? And at that meeting, what I wondered about, it'd be interesting if either states or counties or somebody, maybe industry, would ask for voluntary responses from farmers to say, I use this product and my results were better than my normal treatments. And so it could be turned into a national heat map of, let's say, green was really good for a product and red was bad. But then you could pick any product and see what worked in your region. That would be a huge lift. So I don't know who would do that, but I think your question is really important for what you just asked, Francisco, but then for the other folks on this line, how do we know what works where for what crops?
Yeah, like I said, the Department of Oregon has a very good system where they test for the product labels. They do not get into actually looking at the community compositions, which is the other bit of a sticking point, because you can get things to grow, but are they necessarily what is inside? Because if you use a general media, you're not actually selecting for specific types of organisms. It's just a broad, is stuff in this, are the bugs in the jug actually growing? So I think there is a lot of, and yeah, I think the program you're talking about is a self-selecting um, service, uh, and they don't really talk about how they actually verify their products. And I'd like to see, just personally, a little bit more transparency in that, but something's better than nothing.
Okay, thank you. Dave Huggins has a question too. Oh, you're muted, Dave. Hmm.
Can you hear me now? Sorry.
Yeah, you're good
Yes.
So
now.
just thanks, Tom, for the presentation. As you probably know, there's a lot of interest in the regenerative ag space, and farmers are really looking for, reductions of purchased inputs, et cetera. So this is, a lot of conversations going on right now, and a
Mhm.
lot of testing by growers just trying to see what the heck they can do to reduce some of their input costs. So very timely topic right now. We do intend to just as in response to that to put forward. I'll be supporting a grad student that will be looking at biostimulants. We've got a long list already of interest in terms of what people are talking about mostly, but some guidance along the lines of, okay, under what circumstances would a particular biostimulant really have of a opportunity to be effective? And I think it's a fair question to ask, right, in terms of the sorting out of basically a long list of materials and to focus further our efforts in a productive way in order to perhaps test those ones that are going to have a chance of being successful. So anyway, just wanted to put that out there, and thanks again.
Thanks for that, Dave. Yeah, some of the work that we're doing here in the coastal plain is trying to come up with this circular agricultural economy where a lot of these products, and not necessarily in the biological area, but some of these like frass and biochar, creating a circular economy where it's all, those nutrients stay and are recycled and reused within the area, so we can actually hopefully decrease some of these fertilizer costs by recycling the nutrients and making them available. And in some instances, we're actually seeing that we make them more efficient in their use as they're in the recycled form. So, anything, I think, with the secretary's recent memorandums, decreasing those, improving soil health and then increasing farmer profitability has become a central tenet of our research. And one of the ways we can do that is actually by helping farmers reduce their fertilizer costs, which are a significant burden that they have to bear every year.
Perfect. I think we have 3 minutes for one last question. Christina has her hand up.
Sure. A quick question that I'm wondering if you've seen in cotton. So I'm interested in pathology. And when we see biologicals go in typically to a seed care treatment, usually we see the fungicides taken out. And those are so important in our system, like difenoconazole, for example, for smut and bunt control. So in the cotton system, are you seeing traditional fungicides get removed from the system when biologicals go in? And if so, has that created downstream unintended effects?
Yeah, so we have not, and I think part of the reason is because so all the research that was done across the Cotton Belt was with extension, and I think that they were very hesitant to do seed treatments because of that issue that you raise. And so everything was chosen was a foliar applicant or put into the soil prior to planting. So they did not opt for products that would actually replace or compromise seed protection. So and I don't know of any plans of Cotton Incorporated actually promoting anything that would replace those fungicides.
Thank you.
Hopefully that answers your question. Yep.
Yeah.
Okay, well, thank you, Tom. We're out of time with our meeting. Dave Huggins is leaving us with one last thought. Lots here to say about seed treatments, and actually planting bare seed a bit scary. So with that, thank you very much, Tom, and I'll look forward to seeing you each next year for the Pendleton Science Roundup. So thank you all again.
Bye, everybody. Thank you for your time.